Hello and welcome to our last session dealing with transcription. Before we are going to be talking about the details of transcription, the process, how the information is actually transcribed from the DNA into messenger RNA, we need to have a look at the so-called coding problem. Already Crick, Watson and few others like George Gamow recognized that all DNA is in the nucleus. However, the proteins are made in the cytoplasm. Therefore, they postulated that there must be a genetic messenger transmitting the information from the nucleus to the cytoplasm. And this messenger needs to be able to be decoded into a protein. And this has been partly discovered by the so-called RNA Tie Club in Cambridge. And well, this club was composed of many famous biologists, and it was more or less a team effort. They also postulated that there is a central dogma in molecular biology, and the central dogma is still partly valid until today. And the central dogma claims that DNA gets transcribed into mRNA, messenger RNA, and that this mRNA is then translated into proteins, right? And the central dogma also says that this flow of information is a one-way process. Once you produce mRNA from the DNA, you can't basically go back and produce DNA based on the information from the mRNA. However, what those guys didn't know back then is that there are actually few exceptions of this rule, especially if we think about viruses. Viruses can have DNA or RNA as a carrier of genetic information, and especially viruses that carry RNA are able to reversely transcribe this information in order to produce DNA. However, in normal human cells, the central dogma of molecular biology is still valid. So what do we need to know before we start to look at the transcription? So what are the important concepts and what is the terminology we need to be aware of? First of all, we need to know that the first stage in the expression of genetic information is transcription of the information in the base sequence of DNA to form the base sequence of RNA. Also, only one strand of the DNA molecule, called the template strand, is copied by the RNA polymerase, and this is one of the many differences between transcription and replication. As you should remember from the last session, within the replication, both strands of the DNA are used to produce new DNA molecules. However, transcription, this is not the case. Only one is used. So, because RNA polymerase moves in the 3' to 5' direction along the template strand of the DNA, the RNA product is antiparallel and complementary to the template. I know this is quite hard to imagine, but this is a very important concept, and you should be able to, you must understand this in order to understand transcription. So how does it work? RNA polymerase recognizes start signal on the DNA, and we refer to the start signal as promoters, and we are going to be talking about those structures in detail in our lecture on gene regulation. RNA polymerase also stops at certain points called terminators for each of the 1,000 of transcription units in the genome of a given organism. So what happens first? Again, RNA polymerase locates genes in the DNA by searching for promoter regions. Once those are identified, RNA is able to start, because the promoter is also binding site for so-called transcription factors. Transcription factors are molecules that assist RNA polymerase in their function. Binding of RNA polymerase establishes where transcription begins, which strand of the DNA is used as a template, and in which direction transcription proceeds. In addition to those general concepts, we need to have a look at the strands. So in transcription, we call the DNA coding strand sense plus strand and also refer to it as non-template strand. So this is basically all the same. So don't be confused, because depending on the book or website you might be reading, those sources are going to be using different names. But this is all the same. We have the sense strand, DNA coding strand, and no template strand. In addition, there is also the DNA template strand or antisense strand, and those things are the same as well. Once this is transcribed, the DNA template strand, we end up with the RNA transcript. And this is again anti-parallel and complementary to the DNA template strand. So the DNA here has the following orientation. We have 3' here and 5' here. In the RNA, this is basically the opposite. We have 5' and here and 3' and here. So what are the stages of transcription? The first stage is, as I already mentioned, recognition of the template. So RNA polymerase binds to the DNA. During this step, DNA gets unwound, and this is followed by initiation. During initiation, the process is assisted by certain other proteins you don't need to be aware of, but this is basically the step where the process begins. The next and most important step in transcription is so-called elongation, where RNA polymerase moves along the DNA and synthesizes messenger RNA molecules. Then the unwound region on the DNA moves, followed by termination. Once termination signals are recognized, RNA polymerase reaches the end and gets released, in addition to release of the newly produced RNA molecule. Afterwards, the DNA duplex or double helix reforms again, and transcription is done. Similar to replication, many drugs acting on transcription can be actually prescribed. And first example of a drug which is acting on the transcription is dactinomycin or actinomycin D. This is an integral part of many chemotherapeutic protocols for pediatric tumors. So this drug is used in oncology quite frequently. And it interferes with the elongation stage of the transcription by intercalating DNA, preferentially in the transcription bubble. So once the complex is formed, the drug is able to bind and it prevents transcribing the information. And this stops proliferation of the tumor. Now I would like to show you a video summarizing how transcription works in human cells.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Right. Once a mRNA molecule is produced, this is actually not the end of the story, at least not in human cells, because certain steps need to be done by the cell prior to using such mRNA molecules. And the first step here is mRNA processing. So in contrast to bacteria, DNA in eukaryotes, including humans, contains coding and non-coding regions. And we are calling those coding regions exon and non-coding region introns. So during mRNA processing, those sequences need to be removed. So what you see here is the so-called primary transcript. And the primary transcript gets modified in addition to dealing with the introns. So we have two types of modification of the RNA once the molecules produce. We have the addition of the so-called 5' cap and the 3' poly A tail. The poly A tail consists of multiple adenosines. And in eukaryote, this process, also referred to as polyadenylation, is part that produces mature messenger mRNA for translation. Upon recognition of the AA-UAA signal, the 3' end is removed by an enzyme complex. And another enzyme complex synthesizes a poly A tail and ligates it to the free 3' of the pre-message RNA. Sounds quite complicated, but the only thing that you need to keep in mind is that the addition of the 3' poly A tail is one of the steps in modification of the mRNA, and this is actually helping to stabilize RNA molecules to make them more stable. In addition to this, a 5' cap is added, and this cap is composed of 7-methylguanosine. This also protects from degradation and contributes to an increase of stability of mRNA molecules, and it is required for translation of the mRNA into proteins. As I mentioned before, in humans, DNA contains coding and non-coding region. So during maturation of the mRNA and processing, introns are cut out and the coding regions are spliced together, and we call this process splicing. So, question related to mRNA would be, which one of the following is a post-transcriptional modification of mRNA in eukaryotes? And if you read this question carefully, there is obviously a lot of wrong information provided, and similar to the question I asked you in the last lecture, you should be able to exclude few of those options fairly easy. The correct answer is obviously poly A tailing. Exon splicing is actually wrong because exons are the coding regions and they remain. Also, at the 5' and capping occurs, and vice versa. Tailing happens at the 3' region, so we can exclude those as well. Intron skipping is obviously nonsense, and you end up with only one correct answer, which is poly A tailing. So, why is splicing, removing of those non-coding region, important? Because this can actually result in alternative proteins using the very same DNA fragment. It can also introduce new stop codons, making shorter proteins. It can generate altered reading frames, and we are going to be learning about the reading frames in another lecture dealing with the genetic code. And we can also end up with proteins with different modules or regulatory elements. And a very, very beautiful example of alternative splicing, of basically using the information on the DNA in the most efficient way, is a protein called tropomyosin. What you can see here is the tropomyosin gene at the DNA containing both exons and introns. So we have the coding and non-coding regions. After 3' cleavage and polyadenylation splicing, we end up with actually very, very different proteins depending on the tissue type or cell type those RNAs are produced in. For example, in striated muscle mRNA, we have different coding regions spliced together. In smooth muscle cells, this looks entirely different. In fibroblasts, this is different. And there is also another so-called isoform in brain cells. So this is actually very important because, again, this ensures very high efficacy in terms of using the information contained on the DNA. And indeed, around 60% of human genes have alternative splicing. So the number of proteins that can be produced based on this genetic information is 2 to 3 times the number of genes, resulting in another level of gene regulation, contributing to tissue-specific protein expression patterns. Because, as you can imagine, in neurons, different set of proteins are going to be playing a role compared to, for example, liver cells. And please do keep in mind what we already have learned. Our cells are all different, but the genetic information is the same. So it all comes down to the differential usage of this information. With this, I would like to end up and close the session and the lecture series, and I will see you in our next lecture very soon. Thank you very much for your attention.